All right, it's my pleasure to introduce Lindsey Hartzell. She's a second-year master's student at West Virginia University under the advisement of Dr. Pat Mazik. Lindsey is currently researching non-lethal sampling methods that can be used for fish health assessments by using smallmouth bass. Looking forward to your talk.
Thank you. So today I'm just going to be kind of giving a background of what my thesis is about. I currently don't have any results because I just started doing this. But again, I'm going to be looking at non-lethal sampling methods for fish health assessments by using smallmouth bass. So I'm going to start by just giving a little background information about my project. So first, I'm going to start with smallmouth bass, and they are a freshwater species that were first introduced to the Potomac River watershed in 1854. In streams, these species prefer microhabitats with moderate gradient, low turbidity, and rocky substrate. They're also considered to be a sports fish, and this is because they promote angling for recreational as well as tournament purposes. They're also an indicator of water quality, and this is generally because smallmouth bass do not tolerate pollution very well within their environment. I always like to think of the example that they're kind of like a canary in a coal mine situation, except they're smallmouth bass in a stream. So in the early 2000s, fish kills of different species started showing up, including smallmouth bass. These were recorded in the Potomac River and in the Shenandoah River. They generally took place during the spring season, which is when smallmouth bass are typically spawning. So in 2004, a fish kill happened in the Shenandoah River. And during that fish kill, there was approximately 80% of the mortality was smallmouth bass adults. And when fish health assessments were conducted in the area, it was determined that there was high prevalence of skin lesions, as well as a high prevalence of testicular oocytes. So looking at smallmouth bass in more recent years, and since the first reports of this fish kills, smallmouth bass are still continuing to show signs of adverse health effects. Some of those effects can be parasites and pathogens, intersects, skin lesions, and they can accumulate high levels of certain contaminants. And then there is some areas within the Potomac River drainage that the smallmouth bass populations have been reported to decline, which brings us to lethal sampling could possibly not be the best way to sample fish, since if they are declining, we don't necessarily want to kill more than we have to, which brings me to non-lethal sampling, which is used when euthanasia is not really an option. It's typically more seen with threatened species or endangered species, or like in this case, if a population is in decline in a certain area. That brings me to my own research, which again, I'll be testing out non-lethal sampling methods by collecting blood, mucus, gill snips, and fin rays that prevent injury or death to the smallmouth bass when we collect these samples. I'm also going to try to determine the efficacy of non-lethal sampling methods, as well as successfully age smallmouth bass using fin clips. So I collected samples in May of 24, and we used 3 sites that were in the Shenandoah River and then 1 reference site. So my top 3 sites at the top of the map represent the 3 sites in the Shenandoah, which were the South Fork, the North Fork, and the main stem. And then our reference site was the Maury River, and this was used as a reference site because there has not been a lot of lesions or die-offs reported in this area. So it was a good comparison between the Maury River and the 3 sites in the Shenandoah. And these are just some pictures of the sites that we sampled smallmouth bass at, just to kind of see where we were sampling. So smallmouth bass were collected by boat electroshocking, and we collected 80 fish in total. and this was about, or it was 20 fish per site. So during this collection, these fish were actually euthanized for other fish health assessment research projects, but the sampling methods that I collected could be replicated for non-lethal sampling. So all the fish were weighed and measured. And then we collected for my project blood, gill snips, mucus swabs, and fin samples. And then we also did a skin and gill health assessment index, which was basically we just observed external, if there was any external lesions on the fish, or if there was anything externally we could see with the gills. So with that, I don't have any results, but this is everything that I plan to do. So using whole blood and gill snips, I'm going to look at immune function gene expression. And I think Heather mentioned this in her introduction talk that we'll use the nanostring encounter for this. And then plasma will be used to look at enzymes, protein, and lysozyme analysis, as well as hopefully PFAS analysis. I'm not exactly sure if I will be able to do this, but hopefully we can get this part done. And then blood smears will be used to look for white blood cell type and counts, as well as red blood cell nuclear abnormalities and micronuclei. And then the dorsal and anal fin were collected, and those will be used for age estimation. And since these fish were euthanized for other fish health assessments research, otoliths were taken. So I'm also going to compare the fins age estimate to the otoliths just to see if they're different or similar. And then lastly, I'm going to calculate the condition factor. Okay, so we're hoping with this project that the results of the study can be applicable for future studies involving smallmouth bass and will provide relevant health assessment techniques for other species that are experiencing population declines or have a conserved status. So hopefully my project using these non-lethal sampling methods moving forward can be used in future projects so we don't have to euthanize the fish. So lastly, I would like to thank USGS, the Eastern Ecological Science Center, the Virginia Division of Wildlife Resources for going out and getting these fish for us, and USGS. And then I would also like to thank my committee, Heather Walsh, Vicki Blazer, and Pat Mazik. And with that, I will take any questions.
Thanks a lot, Lindsey. Great talk, and you've got a fair bit of work ahead of you, but it should be exciting.
Yeah.
Hopefully you can come back next summer and present the results.
Yeah.
Folks, if you have questions about the study design or project direction, feel free to unmute yourself and ask Lindsey directly, or you can drop it into the chat. All right, you're getting compliments in the chat if you can't see it yourself, Lindsey, but great job.
Thank you.